Welcome back to the second part of Docker-based analysis. In the previous video, I showed you how to get Docker from the Docker Hub using the command, how to run Docker to create the container, and I'm inside the container of my APRICOT, and we were running few of the commands from APRICOT, which I have commented out here. We use the subcommand query, keyword, select, predict. No, we didn't do with predict. We did until select, where the domains were selected from different databases. Now we will work with remaining of them. For the moment, I'm going to comment out this particular command noscore, which will be covered in different videos. So let's run remaining commands. So CTD-based prediction of the domains in our query proteins have taken place. Now interpro scan is running. So analysis by APRICOT makes sure that if a certain analysis has been already done and the output file is existing, it will skip the prediction of that protein. So if suppose you want to reanalyze it at some point, you have to remove the file created by CDD or interpro from the respective folder. CDD-based analysis is much faster compared to interpro because CDD runs RPS Blast against a single consortium of PSSM database, whereas interpro scan is a collection of different tools. It goes through different databases and therefore it takes time accordingly. Okay, so it's almost done. Now it filtered. This you must be familiar with because we did similar analysis before. What we can add just to challenge our script. Now because we are not running example, we can do real analysis. So I'm going to add one more protein here, which is another E. coli protein, and it has KH domain. So now if I'm giving an input, I have to allow the subcommand query, which is here. Nothing else because we are not changing the second input of keyword. Now let's see how does APRICOT perform. So it took now 3 of them. It downloads 3 proteins information from the database. It's doing the analysis for the one new protein that we just introduced, similarly by interpro scan. Takes some time. It takes time, but as you can see, the runtime is still not that demanding for each protein analysis. Okay, so this is over. All right. So now as you can see, so there was in the beginning only the HFQ, but now after introducing the second protein which has KH domain in it, CDD selects it with several of those domains, and even now interpro has one protein being selected that passes the filtering parameter. So in the next video I will talk about one subcommand which we haven't touched so far in our run script, for example, because it requires embossed needle for one of its annotation. So I will discuss that in next video. Please leave me question and feedback if you have any. If I have missed something which should be covered in a video, I'll be happy to make it for you. Thank you.